Hello and welcome to the SRNA Ask the Expert podcast series, Research Edition. My name is Gigi deFiebre, and I moderated this podcast with Dr. Monique Anderson. SRNA is a nonprofit focused on support, education, and research of rare neuroimmune disorders. You can learn more about us on our website at wearesrna.org. Our 2023 Ask the Expert podcast series is sponsored in part by Horizon Therapeutics, Alexion, AstraZeneca Rare Disease, and UCB. Horizon is focused on the discovery, development, and commercialization of medicines that address critical needs for people impacted by rare autoimmune and severe inflammatory diseases. They apply scientific expertise and courage to bring clinically meaningful therapies to patients. Horizon believes science and compassion must work together to transform lives. Alexion AstraZeneca Rare Disease is a global biopharmaceutical company focused on serving patients with severe and rare disorders through the innovation, development, and commercialization of life-transforming therapeutic products. Their goal is to deliver medical breakthroughs where none currently exist, and they're committed to ensuring that patient perspective and community engagement is always at the forefront of their work. UCB innovates and delivers solutions that make real improvements for people living with severe diseases. They partner with and listen to patients, caregivers, and stakeholders across the healthcare system to identify promising innovations that create valuable health solutions. For this podcast, I was joined by Dr. Monique Anderson. Dr. Monique Anderson is a native of Brooklyn, New York. She received her bachelor's degree in human biology from Stanford University. She attended the University of Virginia School of Medicine, where she earned her PhD and her medical degree, and she completed her residency training in neurology at Emory University School in 2022. Prior to beginning her residency, Dr. Anderson completed the research in support of her PhD in the lab of Dr. Stephen Jacobson at the NIH, where she studied the role of regulatory T cells in HTLV1 associated myelopathies, tropical spastic paraparesis. She was interested in both neurology and infectious diseases, which led to her pursuit of a fellowship in neuroimmunology with Dr. Michael Levy at Mass General. During residency, she worked with and treated patients with MS, NMOSD, MOGAD, and other rare neuroimmune and neuroinfectious diseases. Thank you, Dr. Anderson, for joining me today to talk about your research. To start, do you mind just talking about what transverse myelitis is?
Okay, well, first, thanks for inviting me. So transverse myelitis, really the kind of like the literal meaning is essentially like inflammatory injury of the spinal cord. So essentially this can be due to a number of like reasons, including sort of the immune system inadvertently attacking the spinal cord. It can be due to actually like a virus or some other like infectious agent going after the spinal cord and causing injury. Another potential mode of injury would be where you can have like neoplastics or like cancers that can also cause this type of injury to the spinal cord. And then kind of going from there, other rarer causes such as vascular, as in sort of like if basically, let's say you actually have something that's kind of like attacking the blood vessels within like the spinal cord. So that would be like a vasculitis, and that could also be a potential cause of injury, that similar injury to the spinal cord. But mainly when we're thinking of it, kind of like the two biggest causes are essentially like immune mediated or infectious.
Got it. And so, do you mind just talking a little bit about what you mean by immune mediated versus infectious?
Okay, so they can kind of be, there's a little bit of overlap sometimes. So there are certain, like I'll just kind of pick viruses because those are probably the most frequent. There are certain viruses that have an affinity, let's say for sort of neurons, herpes viruses like in particular, and they can just by themselves, the virus itself can actually cause injury to the structures of the spinal cord. Then basically where you have immune mediated, that can be where essentially it's your own immune system that's attacking areas of the spinal cord. This can actually sometimes be the result or triggered by like other things, whether or not that be like infection as like an example. or it can also just be part of an overall autoimmune disorder. But there's a little bit of kind of overlap even within that kind of like definition. So sometimes we see, and there's certainly like a belief that the immune system can be triggered basically by like stress, viral infections, other types of infections to kind of rev up your immune system to then go and attack some of these structures.
Got it. And then how did you become interested in researching transverse myelitis?
So I actually started off, my graduate degree is from the University of Virginia, but most of my work was actually done at the NIH, the National Institutes of Health up in Bethesda. I worked in the lab of Stephen Jacobson, and that lab is actually focused on both multiple sclerosis, but also a kind of rare disorder called HTLV1 associated myelopathy, tropical spastic paraparesis. So it's kind of a mouthful. It's called HAM-TSP. And this disorder, kind of most of the injury that goes on is within the spinal cord. And I was very interested to sort of like understand why that was the case, sort of like what causes the immune system to target the spinal cord. How does the virus, like, how does it basically sort of like educate the immune system in terms of like moving in that direction, if at all? So all of those questions were a part of like my project at that time. And I kind of became very interested in just any inflammatory injury of the spinal cord. And that's kind of what helped me to figure out what direction to go in for my fellowship.
Got it. And so for your fellowship research, do you mind talking about the study, kind of the background of it, and what led to the development of your particular research project?
I guess back when I was still in residency and trying to figure out which fellowship programs I was applying to, I came across the program at Mass General Brigham, well, I guess at that time, kind of like Mass General Hospital and Brigham Women's Hospital, and they had an MS in Neuroimmunology fellowship. And within that fellowship program, there were a few tracks that you could kind of pursue. And one of them was basically the neuroimmunology track with Dr. Levy. And when I was told that I'd actually get a chance to interview with him during that whole process, I was looking through some of his ongoing research, and I came across the paper that he had written on VPS 37A and a couple of siblings who both had transverse myelitis, and that this was the mutation that was found. And the reason why it kind of became very interesting to me was this was a portion of a pathway that I was actually quite familiar with from my own like prior research. VPS 37A is I'm not gonna, because it's kind of a long name, but it's basically a protein in a pathway that helps to kind of form these extracellular vesicles that I had previously studied. So I'll try to not make this too technical, but basically the S-CART pathway, which is the pathway that this protein is involved in, is called what it actually stands for is the endosomal sorting complex required for transport. And it's a major tool for the recycling and removal of proteins from cells. So this can either help the cell to just kind of like gobble it up, and essentially almost like a trash disposal, and it'll just kind of like be gobbled up within the cell, or it can actually like take those like tagged proteins and dump them into the extracellular space in sort of these extracellular vesicles, some of which are called exosomes. And that was actually, these exosomes were what I'd been studying previously. And so the whole idea of basically proteins that are within the cell, potentially like one, going outside of the cell, and two, that they might actually be getting misdirected from where they're actually supposed to go by mutations, became kind of interesting to me, especially since we're always trying to understand what triggers the immune system to go after the spinal cord, especially like in the case of transverse myelitis. So is it the case of proteins that aren't necessarily supposed to be there actually now being available and present for the immune system to respond to. So that's part of the reason that this whole project kind of like came to be.
Got it. And so, what is this broad research kind of question that the study is attempting to answer? Are you looking at people specifically with, what we describe as idiopathic transverse myelitis with no known cause, or are you looking at people with MOG antibody disease where there maybe is that pathway for why there's inflammation in the spinal cord? Do you mind just talking kind of about that kind of broad research question?
Yeah, so because there's kind of the broad question of transverse myelitis, so we're including both patients with idiopathic transverse myelitis where we do not have a cause, and then we're also including patients with MOG antibody disease, with NMO, with MS, because these are also 3 inflammatory disorders that all involve injury to the spinal cord. And so one, there's the question of whether or not there are any shared like mechanisms between those diseases, but also because for at least NMO and MOGAD or MOG antibody disease, we actually have the antigens or those proteins that we know that the immune system is responding to. And so if we're able to actually like see the presence of these proteins in exosomes, if we're able to see that these proteins in exosomes are able to kind of trigger or inflame, activate that immune response in patients, it might be possible for us to find a similar protein or antigen within the exosomes of idiopathic transverse myelitis patients and see basically like what actually their immune system might have been triggered by or kind of like going after. And then sort of like on the piece of, going back to the whole idea of like that S-CART pathway, how these vesicles were even formed in the first place, if there are similar mutations occurring in the different like patient populations, that might give us a clue of, oh, this might be something that's actually occurring through all sort of like similar diseases. So it gives us an idea of like, one, are there like additional like genetic tests that we should be like doing for like patients? And 2, like, is this a potential therapeutic target for sort of these patients?
Yeah, and I guess in a way, would this then help explain potentially some of those idiopathic transverse myelitis patients, and that the cause then might be more, I guess, explained?
Exactly. And especially because at this point, I mean, at idiopathic pretty much just means that we've kind of like looked at everything else and we haven't been able to figure out like why it's occurred. So if we're able to like further parse out that population and actually see like, oh, actually, even if it's not necessarily VPS 37A, but let's say that we actually find additional components of this like pathway, that there are additional mutations or like variations in those like proteins and genes for those patients, then that's actually like pretty, at least I would find it like very like important, because it's something that we could certainly like look further into and hopefully find therapeutic targets.
Yeah, and as you said, I mean, that's how, people were being diagnosed with transverse myelitis, and then we didn't necessarily know that, about aquaporin 4. And then when the testing for that came about, they kind of got separated into a different category based on that. And so, as you said, parsing out this kind of just big category that we don't really know, that's very, very exciting. And then, as you said, potentially diagnostic and then treatment pathways. So very, very exciting. And so how is this study actually set up? Are you doing testing in actual patients, in cells? How is it set up?
Okay, so I guess I kind of think of it in 2 parts. So part 1 is mostly tissue culture. So within, I guess, like where most people think of like, in the lab or within kind of like under the hoods, working with cells, we have basically a way to express this particular gene, as well as like that gene mutation, and a way to actually knock down or decrease the expression of that like gene within the cells. So that's kind of like the first part, is just kind of looking at cells in culture and seeing what happens with this mutation. Do we see any changes in what the cell looks like, how it functions, getting a better understanding of like what the consequences might be. The other part is we do have actually, and we have continued to collect patient plasma as well as like the cerebral spinal fluid, so the CSF. And we're looking at exosomes, which is one part of kind of like the end of that pathway that I was discussing before, and looking to see what changes we're seeing in the protein contents of those exosomes. Like, are there variations? Are there like shared like proteins, especially like within those idiopathic transverse myelitis patients? Are we seeing anything that's like shared? And if so, like what do we kind of like do with that? So that's part 2 of it. So I guess to answer that, both. So basically like both the most basic stuff, tissue culture, and then also working with like patient materials.
Got it. And where are you getting, are you finding people in the clinic, or is this kind of through other testing that they get done, then you use samples, or how does that work?
So we have patients that had been like recruited previously, like through the clinic, as well as an ongoing, I guess, study where we're able to additionally like ask patients to donate materials. Then there's also, we do have access to biobanks that had previously been established here at Mass General Brigham, where we're able to access certain materials, generally like more so plasma, that had previously been donated by patients.
Got it. And then what is the status of the study at the moment? Are there any kind of results you can report, or is it still too soon?
I have some preliminary results, but I guess like at this point, it's hard for me at this time, I don't necessarily feel comfortable with making conclusions based off of those results. So I'm able, at this point, I've actually been isolating exosomes from patient samples, mostly plasma at this point. I don't have any results yet from like CSF. I have done a few like CSF samples, but not enough to really like say anything. I can say that certainly from the, actually I'm trying to remember, like from the NMO patients that I say that I've actually found the aquaporin for. Again, I think I haven't like had enough patients to be able to say like definitively. I think I'd love to just have a larger number before I actually start saying like, oh yeah, like I'm able to like see this, and I'm able to basically conclude this. But what I can say is that we have dug a little bit further in the patient subsets to see what additional mutations and variations in that pathway I was discussing before, and we actually have found a few, I think it's about 5 other genes within that pathway that shared between transverse myelitis and I believe it was like MS and NMO. So we're going to look at those as well and see basically whether or not they also carry kind of like similar implications as like VPS37A. So I can at least like say that. So I guess what I'd say is that I'd have some preliminary data, but not enough to actually like make any conclusions. I'd feel a lot better once I have added on my numbers. So
Definitely, yeah. And so when do you expect to kind of have, when is the study kind of expected to finish or be at the next iteration of the study?
So I certainly hope that by next summer, fall, that I'll have like the numbers that I want in terms of especially like patient samples to be able to start to actually like really piece together the story. And the same is true on the kind of like the part 1 of it, where like more of the tissue culture based portion, that portion probably a little bit earlier, just because it's not necessarily so much dependent on like the additional like samples like coming in. But yeah, I'd say probably like summer, optimistically, I think next summer.
Great, and we'll definitely check in with you then as well. And so what potential impact does this study have on those diagnosed with transverse myelitis or other rare neuroimmune disorders?
So I think, and the part that I think was most exciting when I was and continue to like do the research in this area is that that same SCART pathway has been implicated in several other neurologic disorders, especially in the realm of repair and recovery. So it's kind of unclear sort of like if that this is also going to be the case with transverse myelitis, but this is something that we're hoping to answer as well. And especially because a lot of my main interest is understanding sort of like what triggers disease and essentially what is that kind of like initiating step. If this does indeed like really determine what antigens or what proteins the immune system may or may not be responding to, again, this could be sort of like something for early intervention.
Got it. And so by repair and recovery, are you saying that the knowledge kind of gained from this might help figure out ways to repair and restore the spinal cord, or
I mean, that
something?
feels lofty.
Yeah.
It feels lofty to state, but I think when I came across that and the fact that this does seem to be a pathway that's been cited again and again with like other neurologic disorders, it has certainly brought up the question is like, well, I mean, especially if it's involved in sort of like the recycling of proteins and the recycling especially of like membrane or like surface proteins, then maybe it actually might have a role in like that recovery and repair, especially like in terms of myelin. If basically like this is something that's, potentially on the surface, you know, can this actually like help with like repairing basically like that damage that has occurred? I don't know. It feel like, but it would be really cool if that were the case.
Yeah, for sure. And then in terms of early intervention, is that just kind of what we talked about earlier with the potential diagnostic piece of being able to identify a genetic mutation or something like that that then could lead to treatments?
Mhm.
Okay, got it. And then anything else that you want to mention that I didn't ask about, or anything else?
Yeah, I think another piece of, and honestly, this is something that's like being pursued by really a lot of other neuroimmunologists, is just trying to figure out not only sort of like what triggers, but what's the difference between monophasic or this happens once versus like this happens again, or this then leads to like progressive disease, or this then leads to basically like additional flares. So that's basically also another portion of this project, is kind of like trying to figure out how do we kind of like put the brakes on things going further.
Got it. Thank you. It's all very exciting, and we appreciate you taking the time to chat with me today about your research and also just doing the research as well and contributing to the knowledge of our community. And so thank you so much.